And good morning, my dear friends. I am Hemanth, and today I am here with a very interesting topic, that is structure and function of cell organelles. So, of cytosol, endoplasmic reticulum, Golgi apparatus, and mitochondria. So, this is the animal cell. In this animal cell, you can see the network-like structure, that is endoplasmic reticulum. And below that endoplasmic reticulum, here you can see another crystal-like structure called Golgi apparatus, and this is mitochondria, and the liquid part of the cell is cytosol. And my question is, what is the difference between cytoplasm and cytosol? Do anybody know? See, cytoplasm means the entire content of the cell, which is without cell membrane and nucleus. Without nucleus and cell membrane, ಒಳಗಿರೋದು ಎಲ್ಲ content is cytoplasm. And without the cell membrane, without the nucleus, without the cellular organelles, only the liquid matrix of the cell is cytosol. Okay, and the cytosol has two types: endoplasm and ectoplasm. Ectoplasm means something outside, endo means inside. That means the peripheral part of the cytosol is called ectoplasm. Andre cell membrane hatra irutte cytosol. The core part of the cytosol is endoplasm. The ectoplasm is very thick and viscous and less granular, whereas the endoplasm is not so thick but more granular. So this is the types of cytosol. In prokaryotic cell, the metabolic activities takes place in the cytosol, whereas in eukaryotic cells, as eukaryotic cells has membrane-bound organelles, the metabolic activities takes place inside the cell organelles as well as inside cytoplasm region also. But in prokaryotic cells, you know that there are no membrane-bound organelles, so metabolic activities here and cytoplasm in prokaryotic organisms. So this is, and the cytosol contains lots of substances like carbohydrates, lipids, proteins, and also many ions, which is responsible for the metabolic activities. And in cytoplasm, there is a process that is taking place, that is glycolysis, breakdown of glucose into pyruvate. This process also takes place in cytoplasmic region. And the next cell organelle, it is endoplasmic reticulum. This is the 3D video that I got through the net. I hope it is more attractive to you. The endoplasmic reticulum, as the name suggests, endo andre ಒಳಗೆ, plasmic within the cell, reticulum andre network. So the word only defined the organelle. The network-like structure which is found inside the cell is endoplasmic reticulum. When the endoplasmic reticulum first observed under electron microscope, this is the microscopic view of endoplasmic reticulum. It looked network-like, so that is why it got its name as endoplasmic reticulum. The endoplasmic reticulum was first coined by a scientist named Porter in 1953. As you all know, endoplasmic reticulum is of two types. One is rough endoplasmic reticulum and smooth endoplasmic reticulum. So let us go to the structure of endoplasmic reticulum. Endoplasmic reticulum is thick as cell membrane. Cell membrane ಎಷ್ಟು thick ಇರುತ್ತೋ ಅಷ್ಟೇ thick ಅದು ಇರುತ್ತೆ, around 50 to 60 angstroms. Then, so the structure of endoplasmic reticulum is of three types. One is cisternae, vesicles, and tubules. So it is also called lamellar, tubular, and vesicular. Okay. So let us see how cisternae looks like. You can see that cisternae are stacked parallel to each other and they are unbranched. The diameter of the cisternae is about 40 to 50 nanometers, micrometers, 40 to 50 micrometers. And in cisternae, you can see ribosomes attached to it. So if the ribosomes attached to cisternae, then what is that endoplasmic reticulum called as? Rough endoplasmic reticulum. So rough endoplasmic reticulum is made of the cisternae structures, and few ribosomes are also attached to the vesicles as well. Then system it also has lot of enzymes which helps in the synthesis and secretion process. Then let us move to the another structure called tubules. Tubules are very branched, network-like, and it lacks ribosomes. Under the microscope, it looks smooth. That means it lacks ribosomes, and this helps in the synthesis of lipid. In a lipid metabolism, tubules part of the endoplasmic reticulum. So then vesicles. Whatever the synthesized protein or any substances which are synthesized in endoplasmic reticulum are transported through this structure, that is vesicles. This size varies from 25 to 200 nanometers, the diameter of vesicles. Then, so we've known what is the structure of endoplasmic reticulum. Let us know what is the function of endoplasmic reticulum. The first function of the endoplasmic reticulum, as we can see in this image, see, ಈ ಇಮೇಜ್ ಅಲ್ಲಿ majority space occupied ಮಾಡಿರೋದು ಯಾವುದು endoplasmic reticulum ಅಲ್ವಾ. So it forms the structure and framework of a cell. It gives support to the cell. That is the first function, and this helps in the transportation, secretion, and lipid secretion, protein secretion takes place in endoplasmic reticulum. This endoplasmic reticulum is also known as intracellular circulatory system. As in our body, how blood are circulated to circulatory system through the circulatory system, even in a cell through the endoplasmic reticulum, the substances are circulated from cell plasma membrane and to the nucleus and other parts of the cell. The endoplasmic reticulum is continuous with the nucleus, cell membrane, and with the Golgi apparatus. The another important function of this endoplasmic reticulum is this helps in biogenesis of membrane. ಇವಾಗ the Golgi apparatus ಗೂ ಒಂದು membrane ಇರುತ್ತೆ, cell membrane ಮತ್ತೆ nuclear envelope, ಇದನ್ನೆಲ್ಲ synthesize ಮಾಡೋಕ್ಕೆ endoplasmic reticulum help ಮಾಡುತ್ತೆ. Especially the rough endoplasmic reticulum helps in the synthesis of nuclear envelope. Then rough endoplasmic reticulum also helps in the synthesis of proteins. They are abundantly seen in pancreatic cells and also intestinal cells and many other parts where proteins are synthesized more. And moving to smooth endoplasmic reticulum. Smooth endoplasmic reticulum helps in the synthesis of lipids. It also helps in the synthesis of bile juice and also steroidal hormones. This smooth endoplasmic reticulum is present abundantly in liver cells. Liver cell allo bile juice secrete agadu. So it is abundantly seen in that cells. Also in many other cells. I have not much went detailed in it. Then endoplasmic reticulum also helps in glycogenolysis. Glycogenolysis, and you know what is glycogen? What the glycogen is? Anyone knows? Can you say? Anyone can say what is that?
Glucose is stored in the body in the form of glycogen.
Yes. Where it is stored? Not in pancreas. The glycogen is stored in liver cells and also in muscle as well. So glycogenolysis andre glycogen is broken down back to glucose. When this happens, when our body is deficient of glucose, ಇವಾಗ ರನ್ನಿಂಗ್ ರೇಸ್ ಚೆನ್ನಾಗಿ ಮಾಡಿರ್ತೀರ, ನೀವು ಬೆಳಗ್ಗೆ ನಾಷ್ಟ ತಿಂದಿರಲ್ಲ, ಬಾಡಿಯಲ್ಲಿ ಗ್ಲೂಕೋಸ್ ತುಂಬ ಕಡಿಮೆ ಆಗಿರುತ್ತೆ. ಮೊದಲು ಜಾಸ್ತಿ ಊಟ ಮಾಡಿರ್ತೀರ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ಆ ಗ್ಲೂಕೋಸ್ ಎಕ್ಸ್ಟ್ರಾ ಇದ್ದಾಗ ಅದು ಗ್ಲೈಕೋಜನ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗಿ ಲಿವರಲ್ಲಿ ಇರುತ್ತೆ. ಆ ಎಕ್ಸ್ಟ್ರಾ ಗ್ಲೈಕೋಜನ್ ಇಸ್ ವಾಪಸ್ ಕನ್ವರ್ಟೆಡ್ ಟು ಗ್ಲೂಕೋಸ್. ಸೊ ದಟ್ ಪ್ರಾಸೆಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಗ್ಲೈಕೋಜೆನೋಲಿಸಿಸ್. Then it helps in glycogenolysis, and then drug detoxification will help endoplasmic reticulum. So next we will go to the next apparatus, next organelle, that is Golgi apparatus. Golgi apparatus was first discovered by Camillo Golgi in 1898, and Golgi apparatus is also known by the name post office of the cell. Right? This is called, see for example, we have something parcel with us and we have to send it to any other state or any other city. We use a courier service like DTDC, Blue Dart, courier service. So this Golgi apparatus in cell acts like a post office Yes, the secreted products which is secreted in the cell reaches to the Golgi apparatus to get modified, to get packaged, and from there it is transferred to other parts of the cell. So Golgi apparatus is also of three types, same as in endoplasmic reticulum, cisternae, tubules and vesicles. So this is the 3D video of Golgi apparatus. Golgi apparatus, in this, the cisternae, which is stacked one above the other, it is sauce-like structure, saucer. You coffee kudikarala that way, saucer. It is like that structure. It is flat and stacked one below the other, parallel to each other. The cisternae part is curved at the margins, and the cisternae is covered by nucleosomes. The structure of the cisternae, which are stacked one another, forms dictyosomes. Then to the peripheral part of the cisternae you can find another structure, that is vesicles and tubules as well. Here you can see the tubules at 1, 2, 3, third cisternae. That is tubular part of the Golgi apparatus. And cisternae is 1 nanometer in diameter, and then we will go to the functions of Golgi apparatus. Golgi apparatus helps in the packaging, modification of the substances which are synthesized in the cell. And another function, a scientist named Burgos in 1955, he found out that the acrosomes in sperm, you know what are acrosomes? Acrosomes are at the tip of the sperm. That acrosomes contains hydrolytic enzymes. Why these hydrolytic enzymes are present in acrosomes? Because when when the sperm fuses with the ova, it should break the walls of the ova, right? So that is why acrosomes has that hydrolytic enzymes which break down the walls of the ova, so that the nucleus present in the sperm enters the ova. So this acrosome organelle is synthesized by Golgi apparatus. And another thing, you know which is the suicidal bag of the cell? Lysosome is also synthesized by our Golgi apparatus. Then Golgi apparatus also helps, I said about dictyosomes, right? These dictyosomes in plants helps in the synthesis of cell wall. It has pectin and other carbohydrate. Which is that other carbohydrate in cell wall? Cellulose. So this Golgi apparatus also helps in the synthesis of cell wall in plant cells. And in plant cells, there are called vacuoles. Vacuoles are a Golgi apparatus which helps in maintenance of fluid levels in a plant cell. So these are few functions of Golgi apparatus. So next, moving on, this is mitochondria. What do you know about mitochondria? What is it called? Yes, it is called powerhouse of the cell. And mitochondria was first coined by, the term mitochondria was first coined by the scientist. Mitochondria, the term was first coined by the scientist Carl Benda. And it is called as powerhouse of the cell, as you all said. Mitochondria's main function is to synthesize ATP. ATP is the energy currency of the cell. So let us go to the structure of the mitochondria. As you can see, you don't see this. See that microscopic view. When you see it in electron microscope, you can see the structure of mitochondria. It may be of cylindrical shape or spherical shape as well. And the number of mitochondria varies in different type of cell. In oocyte, there are 2 lakhs of mitochondria. In unicellular cell, there might be only 1 mitochondria. And the cell requirement, energy requirement, the number of mitochondria may be present. Then we will go to the structure of the mitochondria. In mitochondria, there are 2 membranes. One is outer membrane and inner membrane. The inner membrane has the foldings like this, as you can see. That foldings is called cisternae. the space between inner membrane and outer membrane is called intermembrane space, and the intermembrane space also has the protons which helps in the synthesis of ATP. Then we'll talk about outer membrane. Outer membrane is a permeable membrane which has holes in it. That holes is called porins, which is permeable. Then talking about inner membrane. Inner membrane is not permeable. It is permeable for carbon dioxide, water, and few selected material it permits. It doesn't permit like outer membrane. It has permease enzymes which selects the substance which should enter in or which should go out. Then inner membrane also has many protein complexes and oxysomes, and I'll tell what is its function at the function part. Then inside the inner to the inner membrane, there is a liquid part, that is matrix. That matrix contains circular DNA and ribosomes and many other substances and ions like magnesium, manganese, and calcium. Not calcium, potassium. Then, so we'll see how the mitochondria generates ATP. See, the glucose, or what are all the energy sources for us? Energy source ಯಾವ್ದು ನಮ್ಮ ಬಾಡಿಗೆ? Food is the energy source. From food, there are 3 nutrients that we derive for energy, that is carbohydrate, fat, and proteins. So carbohydrates, after digestion, it is converted to glucose. Proteins are converted into amino acids. Fats are converted into fatty acids. So the glucose pyruvate ge convert agutte. This process is called glycolysis. When the glucose enters the cell membrane in the cytoplasmic region, as I said earlier in cytosol part, there is a process taking place called glycolysis. This glycolysis ಏನು ನಡೆಯುತ್ತೆ ಅಂದರೆ it is I've shown in very simple words. There is an elaborate process in that which I don't know much about it. So how much I know I will explain to you. So the glucose enters the cell. This glucose is converted to pyruvate. This process is called glycolysis, which is happening inside the cytoplasm. And cytoplasmally ಈ process ನಡೆಯಬೇಕಾದ್ರೆ 2 molecule of ATP is secreted or generated. Mitochondriadallu ATP generate ಆಗುತ್ತೆ. Even in the cytoplasmic region, 2 molecules of ATP is generated. So finally, ಈ process ಹೇಳೋ ಅಷ್ಟರಲ್ಲಿ ಎಷ್ಟು molecules of ATP secrete ಆಯಿತು ಅಂತ ನಾನು ಕ್ವೆಶ್ಚನ್ ಕೇಳ್ತೀನಿ, ಅವಾಗ ಆನ್ಸರ್ ಹೇಳ್ಬೇಕು. The 6 molecule of glucose is converted to 3 molecules of pyruvate. The first process. Now, this pyruvate enters the mitochondria. Inside the mitochondria, pyruvate process agi, it releases NADH plus FADH2. ಇದನ್ನು secrete ಮಾಡುತ್ತೆ. ಜೊತೆಗೆ 2 molecules of ATP is also synthesized here. Okay, this cycle is called Krebs cycle or citric acid cycle. ಏನೇನೋ names ಇದೆ. Now, this much ATP is not sufficient for the functioning of cell. ಇನ್ನೂ ATP ಬೇಕು. So what the mitochondria does is, I said NADH plus ಮತ್ತೆ FADH2. ಇದು Krebs cycle ಇಂದ derive ಆಗುತ್ತೆ. ಜೊತೆಗೆ ATP ಮತ್ತೆ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್, ವಾಟರ್, these are the byproducts of the Krebs cycle. ಈ NADH plus ಮತ್ತೆ FADH2 ನಾವು ಉಪಯೋಗಿಸ್ಕೊಂಡು there is another process called oxidative phosphorylation taking place in mitochondria. Inside the matrix, the NADH plus and FADH2 is going into the this is the inner membrane. Okay, this part is matrix. This is the intermembrane space. ಇವಾಗ ಇದರಲ್ಲಿ ಇಷ್ಟು ಚಿಕ್ಕ ಪಾರ್ಟ್ ತಗೊಂಡಿದ್ದೀನಿ ಈ ಇಮೇಜಲ್ಲಿ. You can understand, I guess. This protein is called complex 1. This protein is called complex 2. This is complex 3, complex 4 and complex 5. It is also called as oxygen. The NADH plus and FADH2 is going inside this inner membrane. ಈ ಅದರಲ್ಲಿರೋ ಎಲೆಕ್ಟ್ರಾನ್ಸು ಈ ಇನ್ನರ್ ಮೆಂಬ್ರೇನ್ ಅಲ್ಲಿ ಹೋಗುತ್ತೆ to each of these complexes, and the proton is pumped into intermembrane space. From what? From NADH plus and FADH2. ಅದರಿಂದ ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಇನ್ನರ್ ಮೆಂಬ್ರೇನ್ ಸ್ಪೇಸ್ ಇನ್ನರ್ ಮೆಂಬ್ರೇನ್ ಹೋಗುತ್ತೆ ಮತ್ತೆ ಪ್ರೋಟಾನ್ಸ್ ಗೋಸ್ ಇಂಟು ದ inner membrane space, this space. Okay, so what happens is the protons from these complexes then enters the oxyzone. Here, after that enters the oxyzone has ATPase enzyme. Using that enzyme and binding with inorganic phosphate, 32 molecules of ATP is generated. So in this process, more molecules of ATP is generated. Now, how much molecules of ATP is synthesized only in mitochondria?
34.
Using one mitochondria in a cell, how much ATP is synthesized? 36. Two cytoplasmic regional is synthesis item 34 idrolaಗಿದೆ. So totally 36. Using only one mitochondria. ಅಂತರ ಎಷ್ಟು ಮೈಟೊಕಾಂಡ್ರಿಯಾ ಇರುತ್ತೆ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಇನ್ನೂ ಎಷ್ಟು ಎನರ್ಜಿ ಸೆಕ್ರೇಟ್ ಆಗ್ತದೆ. ಸೊ ಸೀಯಿಂಗ್ ದಿಸ್ ರಿಯಲಿ ಈ ಥರ ಎಲ್ಲ ಪ್ರಾಸೆಸ್ ನಡೆಯುತ್ತಾ ಅಂತ ಅನಿಸುತ್ತೆ. ಲರ್ನಿಂಗ್ ಅಬೌಟ್ ದಿಸ್ ಈಸ್ ಓನ್ಲಿ ವೆರಿ ಇಂಟ್ರೆಸ್ಟಿಂಗ್. ಆ್ಯಂಡ್ ಅನದರ್ ಒನ್ ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ಫ್ಯಾಕ್ಟ್ ಅಬೌಟ್ ಗಾಲ್ಗಿ ಅಪರೇಟಸ್, ಮೈಟೊಕಾಂಡ್ರಿಯಾ ಸಾಕಷ್ಟು ಮುಗಿತು ಮೈಟೊಕಾಂಡ್ರಿಯಾ ಬಗ್ಗೆ. ನಾನು ಅದೊಂದು ಪಾಯಿಂಟ್ ಹೇಳೋದು ಮಿಸ್ ಮಾಡಿದೆ. ಇನ್ ಗಾಲ್ಗಿ ಅಪರೇಟಸ್ ವಾಟ್ ದೇರ್ ಇಸ್ ಅಬೌಟ್ ಆಕ್ಸಿಡೇಟಿವ್ ಸ್ಟ್ರೆಸ್. ಆಕ್ಸಿಡೇಟಿವ್ ಸ್ಟ್ರೆಸ್ ಅಂದರೆ ಇವಾಗ ನಾವು ಇವಾಗ ಆ್ಯಪಲ್, ವೆನ್ ಆ್ಯಪಲ್ ಇಸ್ ಕೆಪ್ಟ್ ಇನ್ ಓಪನ್ ಏರ್, ವಾಟ್ ಹ್ಯಾಪನ್ಸ್ ಟು ಆ್ಯಪಲ್? ಬ್ಲ್ಯಾಕ್ ಆಗುತ್ತಲ್ವಾ? ಯಾಕೆ ಬ್ಲ್ಯಾಕ್ ಆಗುತ್ತೆ? ಆಕ್ಸಿಜನ್ ಅದನ್ನ ಹಂಗೆ ಮಾಡುತ್ತೆ. ಅದೇ ಆ್ಯಪಲ್ ಮೇಲೆ ನಿಂಬೆಹಣ್ಣು ಹಿಡಿದ್ರೆ ಆ ಥರ ಆಗಲ್ಲ, ಕಪ್ಪಾಗಲ್ಲ. ಯಾಕೆ ಆ ಥರ ಆಗಲ್ಲ? Because in air there are free radicals that damages the cells of the apple. That is why it becomes black. But whereas when you add few drops of lemon, so what happens? Lemon has vitamin C, which donates its electron, so that the free radical damage ಆಗದೆ ಇರೋದ್ರಿಂದ ಅದು ಫ್ರೆಶ್ ಆಗಿ ಇರುತ್ತೆ. So even the Golgi apparatus has stored form of vitamin C in it. So oxidative stress, cells ಗೆ oxidative stress ಆಗದೆ ಇರೋಂಗೆ ಈ Golgi apparatus help ಮಾಡುತ್ತೆ. And that's all from me. This is the end. Thank you so much.